I'm Laetitia Denby, and at Waters, I take care of the biopharma application and marketing in Europe. It's my pleasure today to welcome Angelo Palmese. Angelo is the head of characterization and innovative analytics in the biotech development department at Merck in Italy. Hi, Angela.
Hi, Letizia. Thanks for having me.
So, Angelo, you recently participated to a brilliant webinar about the implementation of attribute monitoring analysis for biopharmaceutical development and QC. And in this great work, you shared the way you assessed the analytical technologies and workflow for multiple attribute monitoring assay development, and how you chose BioCore to continue your development efforts and to implement in QC. But can you tell me a little bit more about why Merck chose to implement these attribute monitoring strategies into the biopharmaceutical drug development and QC?
Yeah, indeed. So we started this journey in Merck some years ago, if I'm not wrong, in 2017. So we started discussing about the use of mass spectrometry not only for our characterization activities, but also for process development and then for QC release testing. There are several reasons why we moved towards this kind of approach. So first of all, it's because there are more and more requests from health authorities to, let's say, stick to quality by design principles. And one of the, let's say, most important part of the quality by design is, of course, the capability of monitoring all the critical quality attributes, but not only of our products, so in using state-of-the-art approaches. And so one of the huge advantage of multi-attribute methods is for sure the capability of monitoring in one assay all the quality attributes of the molecule. And this is probably the main reasons why we moved, and we are moving actually from using conventional methods to the use of mass spectrometry multi-attribute method. And then, of course, one of the other reasons is that so we also acknowledge the need of having, let's say, a common platform since early development phase to then the QC release. So in this way, we can, let's say, leverage on the common approach and then on the common modalities also for, let's say, for speeding up the development of the products, and then for using the data we are generating all over the development of our products, also for building the knowledge to be used at release for QC testing, basically. And so last but not least, so here we are talking of a new, let's say, new old method, because in the end, so in reality, MAM is just another way of saying we are using peptide mapping coupled to mass spectrometry. And so last but not least, I was saying, so the advantage here is having one method that is covering a lot of attributes instead of multiple methods. So in this way, we can have a strong rationalization of the control strategy and of the release testing panel. So with a great advantage in terms of lead time for release, but also in terms of added value for the quality of the data we are providing. Because in the end, you can add information to your panel, because with this method you are not only able to monitor your attributes, but also to monitor site-specific attributes that with conventional methods is something not always possible. And then for the BioCore, so I think I went through this during the webinar. So if you are interested in knowing more about that, you can simply have a look at the webinar and attend the next session.
Great. Thank you, Angelo. So yeah, I think that shows how multiple attribute monitoring is growing and how we will develop in the future and allow you to make maybe more safe drugs in a more efficient way and get them quicker onto the market.
Yeah, indeed.
So yeah, thank you, Angelo, and thank you all for watching. And to see more, we invite you to follow the direct link to the webinar that you should find attached to this video.